kind of meetings, especially now. And can you see my screen now? Yes? Okay. So the title of my talk is Miconia as a keystone plant resources for Neotropical frugivores. And as I said, as a disclaimer, I'm not really a melastone guy, but my research is mostly hypothesis oriented. So we use melastone because this is an amazing group of plants. I don't have to explain to you as a way of testing our hypothesis. So to go on resources for frugivores, how to use frugivores. Yes, okay. So the second part, in the first part, I'll show you that Miconia is by far important resource for neotropical frugivores. And then the second part, I will explain why they are so important. And then in the end of the talk, I want to propose a network, a collaborative network, to build a global database on melastone functional traits. And I would like to invite everyone interested to stay here after the talk. Okay, so let's go. Okay, so I want to introduce the concept of keystone species. This is Robert Payne. So more than 50 years ago, he coined the idea of keystone species. This is one of the most powerful and popular concepts in ecology since then. So basically what he did is he experimentally removed this top predator in the shores in Western US, the Pisasterocrassium. And after he removed the starfish, the ecosystem changed dramatically. The California mussel population exploded, and there was drastic changes in the community and the ecosystems. So inspired by the concept of this arc, this small stone here is under the least pressure, but if you remove it, the whole arc collapses. And then he created the concept of keystone species. So that's our species that have disproportionately high impact on the communities relative to their abundance. And traditionally, since 50 years ago, examples of keystone species include mainly top predators, but also some big mammals in the land, in the sea. And those are traditionally being viewed as species that have very high conservation value, because if you remove them from the communities, the whole communities will collapse. But then after a few decades, the concept of keystone species have been widened, have been discussed and evolved. So one of the basically to comprise other species that are not really top predators. So beavers, for example, they have keystone effects on communities through non-food web effects. So build different structures, and this results in dramatic changes in the communities. So the keystone species are those whose effects are large and disproportionately large relative to its abundance. So in order to comprise plants as keystone species, the concept have been redefined, and there have been in the past decades several attempts to identify keystone resources for frugivores. So mistletoes, figs, cecropia, and even melastomes have been proposed to be keystone species because those species, they sustain a large and a wide diversity of frugivores. These papers are really good, very informative. They set the basic stones for identification of keystone species, but there's a problem here because all these species, all these studies, they have a local limitation and temporal limitation. So most of them are related to people going to the environment, observing frugivores eating fruits, and then for a limited part of the year, a limited region, and then you have no idea on how these plants compare to each other because they generally focus on a given group. So in order to find broad scales of keystone species, we need a different approach. And what we did in the studies are to use fruit frugivore networks, trying to assess the role of Neotropical keystone species. So the only way that we can assess the role of keystone species is to remove them from the ecosystem or to include them in the ecosystems and observe the cascading effects on the community stability and diversity. Doing that is nowadays is kind of unethical. You cannot go to the woods and try to kill all the elephants and see what happens to a given community, or kill all the lions or jaguars. So one way that we can really assess the role of keystone species is doing simulated species removal from networks. So I don't know how many of you are familiar with ecological networks. This basic representation of how plants, the squares, interact with frugivores, the squares. So every time you see a bird or a frugivore eating a fruit, then that's a positive interaction, then you score 1. And when this interaction is not present, then you score 0. So you can imagine that if you remove this kind of plant here from the community, you have a much larger effect on the community than when compared to the removal of these species which interact with only one frugivore. So we hypothesize that keystone species in the networks promote community stability. Then if we remove them, this will result in higher change in the network structure, then higher than expected by chance. So what we did here is to assemble 38 fruit frugivore networks across the neotropics. You can see here that we sample from almost the Tropic of Capricorn to the Cancer, including Galapagos on islands, from the sea level to the Andes in Western South America. We have more than 6,000 interactions, almost 1,200 species of plants across a wide diversity of families, and we included 31 networks from forests and 7 from open ecosystems. So we basically cover the wide diversity of ecosystems and geographic locations in South America. And we have to develop a new approach to identify these keystone resources. So what we did here, we have this matrix that represents the interactions between birds on the column, plants on the rows. So every time you see 1 is an interaction that has been observed, and 0 is an interaction that was not observed. So we had 38 matrices of these ones, and we calculated simple metrics here, what are called normalized degree, centralized centrality, and between centrality. For those who are not familiar with these terms, these are basic descriptors of the network who inform which species are more connected. So the higher values of each of these 3 metrics here show which species are more connected in the network. So for each of these 3 metrics, we calculated the first quartile, so the top ones which have this centrality measures here, those species that are more connected in the network. And then for each individual metric, so centralized, centrality, normalized degree, and between a degree, we just made a threshold, a cut, and then we selected the 10 top for each one of them. And then the final step was overlapping all those 3 metrics. And the species that, the genus or the genera that overlap into these 3 metrics would be identified or retrieved as the potential keystone land resources. So I will show the first results here. As you can see, for the 3 metrics, Miconia is by far the most important resource for frugivores here. So Cecropia and Ficus are often shown, even in textbooks, to be important keystone resources for neotropical and even paleotropical fauna here. But then, as you can see, Miconia wins by some orders of magnitude here. And when we overlap all those 3 criteria, then Miconia is, as you would expect, the top one. So from the 108 families that we have here with a lot of genera, we have only 6 genera which have been retrieved as keystone resources. So are the genera that are the top 10 across those three criteria here. So Miconia, Cecropia, Ficus, Cecropia, Clusia, and Bersonia. Some of those have never been argued to be keystone resources before. So, and then our second approach was to evaluate the effects of removal simulations on the network properties. So as I told before, we cannot go to the field and remove the species. So what we can do is simulate their removal in the networks. And we tested the effect of removal on three metrics: nestedness, modularity, and niche overlap. So nestedness is a property of networks where less connected species are just a subset of the more connected species. So so you can see the bird species here, you can see the plant species here. We even found clip art of Miconia to use here, and we hypothesized that the keystone species are shown here in red would have higher nestedness, they would have higher modularity, and then the niche overlap here. So this is just a graphic representation of a network, a nested network would look like. This is a representation of a modular network. Modularity is just a property of the networks, which we have some subgroups of species which interact within their group more than species from the group. And then niche overlap are species that connect different species, but they have overlap with other species as well. And this is a graphic representation. So what we did here is remove some allostomes, observe the effect of nestedness, of modularity, and niche overlap. And then we have removed randomly some other species from the network to compare the effects of Miconia removal, not only Miconia, but the 6 genera, and then random removal of other species. We selected nestedness, modularity, and niche overlap because these have been shown as surrogates of community stability, resistance to disturbance, and species loss. So changes in nestedness, modularity, and niche overlap would mean that the communities are more prone to destabilization. And this is basically what we found here. So we have the 6 genera for nestedness, modularity, and niche overlap. And we have here the removal of each of these genera, the effect of removal on the nestedness, and the removal of random species. That's our new model. And then I highlighted here, you don't have to read all the table, but we can see for the 6 genera, for nestedness, modularity, and niche overlap, we have a statistical value here shown in bold, and I put the red arrow to indicate where you have significant effects. And as you can see, as a general, when you remove Miconia from a community, then you have a significant decrease in nestedness, but for other genera, except Cecropia, this does not exist. So for Miconia and Cecropia, yes, when you remove them from the communities, nestedness changes. For modularity, only the removal of Miconia was significant. And for niche overlap, okay, for Miconia, Cecropia, and Bersonima, when we remove those species, the niche overlap changes. There's a decrease in niche overlap. So basically what we show here is that across all metrics, Miconia stands out as the most important species creating community stability and resilience to disturbances. This is, it's more complex than that, but basically I don't want to show the details. This paper has just been published last month in Biotropica, and all the credits go to João, who's here in the room, and Tadeo, and Wesley from Mexico as well. So this guy here was his master thesis. He's a brilliant student. He's now a PhD student at the Pennsylvania State University. And you will hear more about João in a minute, okay? So now that we show that Miconia is really the most important group of plants sustaining frugivores across the Neotropics, we wanted to know why they are so important. So we have reviewed the natural history of Miconia. It's a big group, you all know that. And then we try to get the functional traits that are related to keystoniness of Miconia. So in order to do that, we compile data from fruit traits and fruiting phenology. We integrated the scattered information on frugivore in Miconia to provide the first assessment of the number of animals that eat Miconia fruits, and I think you'll be surprised to hear that. And we also, finally, we evaluated the effects of gut passage on the germination of Miconia species using meta-analysis. So we have a data set for fruit traits of 360 species, for fruiting phenology, different species. As you can see here, the number of species is different for different traits because this information is really scattered in the literature, so it's just like mining all the data from the literature. It's really time consuming, and all the credits again go to João for doing that. And we have data spread across the Neotropics, or more sites than we have for the Netherlands. But as you can see here, our data set is far from being complete. And for that passage, we have 35 species. Okay, so this is a rows diagram showing the number of Miconia species fruiting every month in the Neotropics. So this is January, February, and so on. So this is the result of statistical tests, which tests whether the distribution is different from random. As you can see, the p-values are non-significant. This means that the plants are fruiting year-round across the Neotropics. So this shows that Miconia species fruit all over the year. But of course, you have to go into different communities and look what is going on there. And in the Neotropics, we have these moist habitats, the seasonal ecosystems, and we have the seasonal habitats where there's a period of the year where it doesn't rain too much. So this is just one example of a seasonal ecosystem where there's the dry season here. As you can see, no matter when you go to the field, there will always be one species of Miconia fruiting. No matter when, there will always be some Miconia fruiting. And if you go to a seasonal ecosystem, you also see that doesn't matter when you go there, there will always be some Miconia fruiting as well. So Miconias fruit year-round on the Neotropic. If you're a frugivore, especially if you're a frugivore bird, you always can rely on Miconia for food. So this is the histograms showing the distribution of different fruit traits of Miconia. And just to sum up here, Miconia fruits are small, as we all know. The seeds are also small, although there is some variation. And there are kind of large number of seeds per fruit, but this is really outstanding. And the fruit crop size of Miconia is something that's almost unbelievable. Hundreds of thousands of species of fruits in a single individual trait. So this is another point that makes Miconia reliable for frugivores. So small fruits, which have high content of water and sugar, which makes them very easy to digest, and they are produced in high amounts. So frugivores can really rely on these resources. Half of the Miconia fruits are black, but also purple and dark. As you can see here, there is a wide diversity, but those are the main frugivores. And early studies in the 90s by people studying in the Central America by Julie Denslow, that these Miconia fruits are basically bird dispersed. But as we can see here, and studies have shown in the past decade, many other frugivores also rely on melastonic frugivores. As you can see here, birds, they can eat all kinds of colors, and mammals as well, but reptiles, they kind of prefer purple fruits. We don't know if it's a pattern because the sample size here is really small, but it seems that lizards and turtles, they prefer purple fruits. Ants also don't care about fruit color, so all fruits that fall to the ground in the savannas, in the forest, they can be removed by ants, no matter what color they are. And that's what we did. So we want to estimate the diversity of frugivores eating Miconia food. So this is basically a rarefaction curve showing the number of species that consume Miconia foods. And this on the x-axis is the number of studies of Miconia species. So we can calculate and estimate the number of species that can eat fruits using this as sampling units. And you can see the shadows are confidence intervals. And you can see the small Miconia studies are the species richness increases for birds, increases for ants, for primates or mammals, lizards, and even fish. This is inset here is just a highlight of this minute part here of the graphic. As you can see, none of the curves stabilize or reach an asymptote. So this means that diversity of frugivores is still underestimated. We found 600 and almost 50 species of animals consuming Miconia foods, but this is certainly an underestimation because the shape of, so if you add more species, the curves will keep growing. And this is unbelievable for me. If you look at Miconia albicans, this is my favorite plant, 122 species of animals consume its fruits. This is really outstanding. I don't know if any of you would know 122 bird names, but this Miconia albicans is consumed by all of these plants. And you can see here, this is a widespread species, so it has the opportunity to be in contact with different frugivores. And this is Miconia ipoleuca, a fourth species that has been argued to be a keystone resource in the Atlantic forest. But this is just a opportunistic study where researchers have counted the number of birds eating its fruits when a gap opened and then the plant fruited. So there is a very limited temporal assessment here as well. But then you can see lots of birds also, they don't care about which Miconia, they eat lots of Miconians as well. And this is really a little bit strange because it has been argued that some fruit, especially green fruits, are not visited by a high number of birds because the role of green fruits is to photosynthesize. And this has been shown 40 years ago. But this Miconia albicans produces green fruits and is the most consumed plant species probably in the world. And lastly, we aimed at understanding the effect of gut passage, not only gut passage, but manipulation by ants across different groups of vertebrates and invertebrates on the seed germination. And we did this using meta-analysis. So this dashed line here is showing where there is no effect on the gut passage. If you move to the right side, this shows a positive effect, a proportional change relative to the control. So values around here means that germination increased by 100%. And if we move to the left side, values around here show that germination decreased by 100%. These are the confidence intervals, and if they overlap with the dashed line, this means no significant effects. So we had enough data to compare manipulation by ants and gut passage by rodents, Didelphimorphia, that's possums, primates, and birds. We have detail on different families and taxa within these groups, but I'm just going to show you a general pattern here. And when ants, they collect and if Miconia fruits fall into the ground, they decrease germination significantly by almost 100%. No significant effects by rodents. I'm really surprised is that a really positive increase by possums, a decrease by primates and birds as well. Birds was really a surprise to us because melastomes have been known for decades to be bird dispersed fruits, So we expected that coevolution might lead to positive effects, but birds actually decrease seed germination in Miconia. This is something really unexpected. And the explanation for that is that, I mean, our explanation for that is that if you can look at the percentage of germination across different groups here, we have to compare not only with the hand extracted seeds, but we also need to include this additional treatment, which is germination within the fruits. So when fruits are not eaten in nature, there's no one to remove them. There's no one to remove the seeds from the fruits. So they remain there and they don't germinate within the fruits. Firstly, because Miconia fruits, they have germination inhibitors in the fruit pulp. They prevent germination. They also have light demanding seeds. So if they are within the fruits, light cannot reach the seeds and they won't germinate. And the fruits are basically water and sugar. If they are not consumed by frugivores, they will rot, they will be attacked by fungi, and fungi will kill the seeds. In the laboratory, people extract the seeds, really set them to germinate, and then that would be a more honest comparison or more reliable comparison with the effect of different frugivores or seed dispersers. And then as you can see here, if we compare the germination of hand extracted seeds with that of different frugivores, then you can see that birds and primates are not doing that bad. Because the main service provided by these frugivores is not scarification of the seed testa, but rather seed cleaning, the removal of the pulp from the seeds and dispersal away from the mother plants. And still, the ants, they provide a less significant service, but if you compare the germination of ant manipulated seeds with that from seeds within intact fruits, you can see that ants still provide reliable and positive services. So to conclude my talk, I would like to say that Miconia is a key or top keystone resources for neotropical frugivores. Its fruits are small, water and sugar rich, and available year-round, so they are reliable source biodiversity of frugivores spanning basically all lineages of vertebrates, excluding the amphibians. So the gut passages are still not clear, maybe hard to explain, but the main service it provides by frugivores is the seed cleaning or the pulping and the movement of seeds away from the parent plants where they can find safe sites for establishment. And we predict that Miconia fruits will be even more important for neotropical frugivores in a deformated and fragmented forest in the future. So we are experiencing a significant forest fragmentation and habitat fragmentation. So you all know that Miconia species tend to be pioneers. They tend to do well in degraded ecosystems, in fragmented sites, in forest borders and road edges. And we are also experiencing significant decreases in the size of seed dispersers and in the size of fruits in neotropical forests. Defaunation has resulted in the killing of the large-bodied frugivores and seed dispersers, and the killing also in their associated large species with large seeds. So the species with large seeds are decreasing together with the decrease of large species of frugivores. So I would like to thank very much João and Tadeu. They deserve most of the credit for this, but also I would like to thank Wesley for being author in the first paper, and my best friend Tatiana, I think she's also here, and Lika, my former students. They have been involved in the second paper, which is now in anosopotamy, in minor review. So I think we are almost there. And those people, this wonderful team, is basically responsible for all the credits in this presentation here. With that, I would like to thank everyone for the presence, the funding agencies, the game study group, who supports me a lot. And I'm happy to take any questions from the audience. And after that, I will be very pleased if we can discuss the idea on the trait database. Thank you very much, everyone.
Nice. Fernando, thank you for the amazing talk. All this information about Miconia is really interesting. So let's start with the questions. There's one here, Virginia Paixão. She's asking, all those animals offer primary seed dispersal service, or are you considering also when fruits are consumed in the ground?
So in the second paper, I suppose, we consider both primary and secondary seed dispersers. So we sample data, but as you can see in one of my pictures, even ants, which are known to be secondary seed dispersers, they can be primary seed dispersers as well. I showed the picture where Atta, it climbs to Miconia albicans shrub and removes the fruit. So it's a primary seed disperser. But in this paper, we have considered all primary and secondary seed dispersers as well. So we sum up everything to provide a broad overview of the total number of seed dispersers.
Okay. So we have another one, Agnes. Is there any work being done on the impact of different fruit dispersers or diversification in Miconia?
Not that I know of. I know that there's a recent paper, I think by Marcelo Reginato and colleagues, I suppose he also is here, he can speak better than I, showing that, attempting to show whether Miconia fruits have been in a key innovation for the diversification on the clay, but I think they didn't find any significant results showing that, to the idea that Miconia, these small berries are a key innovation. Not that I know of in Agnes. So there has been some studies showing that defaunation can lead to changes in fruit traits, and this could be related to changes in diversification. But most of these studies are concentrated in palms, which have been argued to be keystone resources as well, but not on a neotropical scale according to our results.
Okay, thanks. We have another one from Julie Denslow. I wonder how interchangeable fruit resources are in tropical forest. If frugivorous birds consume a wide variety of fruits in different families, shouldn't the keystone resource be small sugar fruits rather than particular taxonomic groups?
Good question, Julie, and thanks for inspiring our research on the seed dispersal in melostomes. So yes, we had, as I showed in the paper, we had 108 families. Other families as well should also produce water and sugar-rich fruits, but they were not retrieved as important keystone resources on the neotropical scale. I think it's important to mention that our first study identified keystones on the neotropical scale, on a continental scale. This doesn't mean that other groups of plants are not keystone species on a community level. So we're not saying that palms and cecropia and ficus and other small berries are not important for frugivores. They are on a community level, but perhaps on a neotropical scale, when you look across the continent, they're not as important as assumed to be previously. So that would be my answer to this question. I don't know if it's enough, Julie.
Thanks, Fernando. So Agnes has another question. So do you see any ecosystem pattern? For example, are Miconia fruits more abundant or important in lowland important in lowland rainforests than the high elevations?
Amazing question, Agnes. We have included elevation and latitude in our models in the first study, to see whether the relative importance of the different keystone genera changes with latitude and elevation. And we have found, and we were happy to find non-significant results for all results. This is when you're happy to find non-significant results. And this shows that across the whole latitudinal elevational gradient, our results stand the same. This means that the genera are the same across the whole elevational and latitudinal gradients. So Miconians, Ficus, and Cecropia, they stand as important keystone resources no matter where you are in the Neotropics.
Okay, thank you. So we have from Lucas Majur. It would be interesting to break this up into different forest types as well. As for instance, Miconia are not very prevalent in seasonally dry tropical forests across the Neotropics, whereas other dry adapted taxa would be. So.
Yes, so Lucas, in our database, we didn't have any network from tropical dry forests. You're right, they are not really abundant and diverse in tropical dry forests, so we didn't have any network on tropical dry forests so that we can include this covariate. We had 38 networks, 31 from wet forests and 7 from open ecosystems, including savannas, grasslands, wetlands, but 31 versus 7 is not enough sample size to make comparisons, statistical comparisons. But I suppose that even in open seasonal ecosystems, they also stand as important keystone species as well.
So everyone is on mute now. If you want to have to do a question, please unmute the microphone so you can ask directly to Fernando. So.
Fernando, great talk. I was wondering, so can you somehow measure not only the number of species, but also what is the proportion? So in a given site, you have ficus, you have Cecropia, you have Miconia. Can you measure the biomass proportion of the importance of the different groups, or is that too complicated?
That's too complicated. With the data set that we had, we didn't have, it was difficult to get enough data to do this analysis, let alone do this that you are suggesting. I think the key idea of the keystone resources implies that species have low abundance and high effects on the ecosystem. One of the shortfalls in the limitations of our study is that we don't have any data on species abundance within the community. So it might be only that we have like a Miconia dominated ecosystem, and then of course by chance birds and mammals would eat more Miconians than you would expect. This is just a random process, but this information is really not available in the literature. But I know of global initiatives which are now mapping abundance within the community. So they have plots with species abundance within each community. And then if you can overlap that with the frugivore networks, then that will be a dream to come true, to then do exactly what you're suggesting for them. This would be a next step, really advancement.
Carmen have a question. So. Fabio, can you mute Carmen?
If she can now. See if she can. Carmen, can you try?
Okay, yeah. Hi, Fernando. Very nice talk. I was just wondering, all of your sites, if I remember correctly from the first map, are in the lowlands and none are in the high Andes, really? Is that correct?
No, we have a few sites in the Andes too.
Oh, okay.
But we have one. I have some up to 2,500 meters above sea level.
Okay, it would be interesting because Miconia can go above 3,000 meters. So, you know, just to see what happens up there, because the fauna, of course, is different up there as well. But really interesting. Thank you.
Yes, I've seen some Miconia in the Páramos in Colombia, almost 3,000 meters.
Yeah.
It was really similar to Miconia because really catch my attention. It was amazing. But I couldn't see any bird eating fruits there. But for sure, the problem coming, we haven't found any significant effect of elevation, and this supports the idea that Miconia is important also up there in the mountains. But we didn't have plots established along the gradients, so it would be really important to have a gradient of networks along the elevation gradient. They are not evenly distributed. This would be really great to take a look at.
Yeah, that would be great. Thank you, Fernando.
Yes. Thank you, Carmen.
Hi, Lele.
Oh, Vini.
Thank you for your amazing talk. I may ask a very silly question. I just want to know your opinion. If the Miconias, these, how can I say, if the prevalence of apomixis in this genus can be linked to their importance, or, you know, to their importance as fruits to frugivores.
Yes. Vinny, that's far from being a silly question. It's a very interesting question. I don't have an answer for that. But we can take a look at the most important taxa of Miconia, the most important species, and I'm sure you have the database on the apomictic species, and we can just contrast and see whether there is an effect. This would be a really nice thing to look at. João is here, just by your side on my screen, and he has all the database. And one thing that we want to do, people, is that we want to make all database freely available for everyone. So anyone can use and test wherever variables, wherever you want to test. So that's the purpose of the network that I want to discuss with you guys. But that's a very nice question, Vinny. We should take a look at this, or maybe you should take a look at these things. And for those who don't know me, basically no one calls me Fernando, it's Lele. So it's really strange that people call me Fernando. Okay, thanks, Vinny.
Okay, Marcelo has a question.
We have to unmute him.
Okay, so nice talk, Fernando. I also have, I guess this is also a silly question. I'm really glad that Miconia is like the keystone, the top keystone, but isn't that related to the size of the genus somehow? A little biased because of the size?
Amazing question, Marcelo. We also wanted Miconia to be the first, and we were really happy to see that. And then this paper went for review in seven journals, and then we managed to finally publish this in Biotropical. But then reviewers criticized that, and we also looked at the genus size when we compared different keystone species, and we made a regression looking at the values of keystoniness and by the number of species in the genus, and we found no significant relationship. So this means that it's probably not related to the genus size as people would expect as well.
Okay, cool.
But this makes sense, yes. So yeah, we found, for example, Solanum is larger, but doesn't even appear.
Okay, so does anyone have more questions? Now is the time.
Okay, so it seems that Marcelo wants to ask another question, or is it no? Yes or no? You're
Yes.
good. Okay, go ahead, Marcelo.
Are you listening? So I wonder how are you about your database that you're trying to compile? How is your approach on doing that? It's like literature mining.
Yes, we had some keywords, and then we had to retrieve all the papers, scan the abstracts to see whether there is any Miconia species in the paper, and then we had to read all the. I mean, not me, but João, who is here. He was responsible for reading all the papers and mine all the data from the literature. So he spent months, probably a year or more, doing this, and then we get everything from the literature. And when the data was not readily available, we emailed the authors, the corresponding authors. We asked for the data. They provided, so we can say that we have a relatively good database for the moment. But I suppose that you guys, the taxonomists, the specialists, are the ones who have more data. So that's why I want to discuss the database approach with you guys.
So manually, right? If I understood.
Sorry?
It was like manually. The guy was picking manually, right?
Yes.
Okay.
That was his master thesis.
Okay.
So he spent 2 years doing that.
So I think that's I'm gonna stop recording now, and I think